with feedback loops, and their main function is to coordinate or synchronize the output processes to the external environment of the plant so that they may function in a time-dependent manner. Now, among the many signals being perceived by the input components, light is often being considered as the most informative signal for plants because light is also used by plants as an energy source to optimize their growth. Now, the perception of light by plants is mainly mediated by the proteins called photoreceptors. And among these proteins, phytochromes and cryptochromes are well known for their ability to influence the clock genes within the plant circadian system and to also control time-dependent output processes. However, aside from phytochromes and cryptochromes, another prominent set of plant photoreceptors are the blue light photoreceptors called phototropins, which are mainly involved in photosynthesis. So previous reports have already suggested that these phototropin receptors do not affect the clock genes within the plant circadian system. Having said that, certain aspects of the phototropin-mediated photosynthetic process have been shown to demonstrate rhythms in plants, which may suggest that these processes are clock-regulated. However, again, the lack of reports stating phototropins as possible input components of the circadian clock have made it difficult to understand these findings and to also establish a connection between clock and phototropin signaling. Now, with previous reports stating that the PRR clock genes are significantly expressed within the mesophyll cells, which are the primary sites of the photosynthetic process, and that they can enhance daily photosynthetic activity by regulating sucrose synthesis and chloroplast gene expression, it would be reasonable to speculate that the PRR clock genes are somehow involved in the phototropin signal transduction pathway. So with this idea in mind, we articulated the following hypothesis for this research, that phototropins control the photosynthetic process through their regulation by the PRR clock genes. So to verify this hypothesis, we designed experiments that would answer the following questions. First, do the PRRs affect phototropin-mediated responses? And secondly, what is the potential regulatory mechanism? What is the potential regulatory mechanism between PRRs and phototropin signaling? So to answer these questions and to somehow elucidate the potential connection between PRRs and phototropins, we first determined the effect of the PRRs on chloroplast movement in both wild type and PRR misexpressing lines. And then secondly, to identify the potential regulatory mechanism between PRRs and phototropin signaling, we specifically analyzed the gene expression patterns as well as the phosphorylation status of the phototropin receptors under blue light conditions. And then finally, to fully establish the link between circadian clock and phototropins, we also investigated the possible time dependence of the chloroplast movement process. So for the first objective, chloroplast movement was specifically analyzed by performing confocal microscopy. In addition, chloroplast movement was also quantified by determining the percent chloroplast coverage, which was computed by dividing the total area covered by the chloroplast over the whole cell surface area. And after analysis, our results revealed that at dawn or early morning conditions, chloroplasts tend to settle at the bottom of the mesophyll cells, or the dark response. Upon excitation of blue light, chloroplasts gradually accumulate on top of the mesophyll cells for light absorption. And then under strong blue light intensities, chloroplasts move towards the sides to avoid photosynthetic damage. Now, relative to the wild type plant, in the PRR 579 triple mutant, we were able to observe that the dark response is normal during early morning conditions. However, under weak blue light conditions, the triple mutant is obviously showing a higher rate of accumulation response. And under strong blue light intensities, we can clearly see here that the avoidance response is severely attenuated. Finally, for the PRR7 overexpression line, we can see here that under all light conditions, this particular line is demonstrating attenuated movements. And actually, what we're seeing is that the chloroplasts tend to constantly be positioned at the sides of the mesophyll cells. So when these confocal images were quantified, again, under weak blue light conditions, where the chloroplasts are expected to demonstrate the accumulation response, in the wild-type plant, the area being covered by the chloroplasts gradually increases, which may suggest that the accumulation response is successful in the positive control. Similarly, in the PRR579 triple mutant, the area being covered by the chloroplast also increases. However, it is obviously at a higher rate when compared to the wild type. Finally, in the PRR7 overexpression line, this line continued to demonstrate low chloroplast coverage values, which may suggest that the accumulation response is severely attenuated for this line. Next, under strong blue light intensities, where the chloroplasts are expected to demonstrate the avoidance response, the wild type continued to demonstrate low chloroplast coverage values, which may be corresponded to the chloroplasts always being positioned at the sides of the mesophyll cells. Similarly, the PRR7 overexpression line also demonstrated low chloroplast coverage values, which are comparable with the wild type. Conversely, the PRR579 triple mutant still is demonstrating higher chloroplast coverage values even under strong blue light conditions, which again may suggest that the avoidance response is attenuated. So taken together, these results here may suggest that the PRRs are regulating chloroplast movement, specifically by inhibiting the accumulation response under different blue light conditions. Now, since our results here show that this phenotype is strongly influenced by the PRRs, it would now be prudent to investigate how the PRRs actually impact the genetic mechanisms regulating this specific phenotype. So in that regard, a time course analysis of phototropin expression was done, and our results revealed that the transcript levels of both phototropin 1 and phototropin 2 were upregulated in the PRR579 triple mutant, while relatively lower transcript levels of phototropin 1 can be observed in the PRR7 overexpression line when compared to the wild type. In addition, the same results were kind of observed at the protein levels of the phototropin receptors, which may suggest that the PRRs are negatively regulating phototropin expression, and that this regulation consequently influences the concentration of the phototropin proteins or of the phototropins at their functional protein levels. So based on these observations, it would also be very informative to know whether this transcriptional regulation happens directly or indirectly between PRRs and phototropin receptors. So by performing CHIP qPCR analysis, we were able to observe that the PRR7 protein directly regulates phototropin expression, specifically by binding onto the upstream regions of the phototropin 1 and phototropin 2 DNA. In addition, the lack of binding signals from the TA3 gene, which serves as the negative control for this analysis, may suggest a specificity of binding between the PRRs and the phototropin receptors. Now, aside from phototropin expression, the results of our Fostak gel Western blot analysis also revealed that the PRRs are influencing the phosphorylation status of the phototropin 1 receptor. More specifically, we can observe here that relative to the wild-type plant, under weak blue light conditions, the 579 triple mutant is demonstrating faster rates of phosphorylation, with phototropin 1 being phosphorylated after shorter periods of weak blue light exposure. So this result here may suggest that the PRRs have more significant impact towards phototropin 1 activity by specifically influencing both expression levels and phosphorylation status of this receptor under weak blue light conditions. So finally, given that our current findings strongly suggest that phototropins and chloroplast movement are tightly associated with the circadian clock, for the final objective of this research, we investigated the possible time dependency of the chloroplast movement process. And for us to do this, we basically compared accumulation response happening at ZT0, which simulates morning conditions, and at ZT16, which is a time point that simulates evening conditions. After analysis, our results revealed that in the wild-type plant, the accumulation response happening at ZT0 or at morning conditions occurs at a higher rate when compared to the accumulation response happening at nighttime conditions. In addition, the PRR5793 triple mutant still demonstrated higher rates of accumulation response, while the PRR7 overexpression line continued to display attenuated movements at both time points. So based on these observations, the results being demonstrated by the wild type at ZT0 and ZT16 may suggest that the accumulation response may be working in a time-dependent manner, and that the PRRs actually exert regulatory control over this time-dependent nature of this chloroplast movement. So to summarize our results, our findings include the following. So the PRRs are involved in the proper regulation of chloroplast movement. PRRs regulate phototropin expression, as well as the phosphorylation status of phototropin 1. And finally, chloroplast accumulation response is working in a time-dependent manner due to the influence of the PRRs toward phototropin 1 activity. So with these findings, I think we can safely conclude that the PRRs, they somehow contribute to the enhancement of photosynthetic efficiency by regulating phototropin-mediated responses such as chloroplast movement. So before wrapping up, I would like to acknowledge first the members of our laboratory, my supervisors, our collaborators from different universities, and of course, the institutions that are continuously supporting my research. So that's it for my presentation, and thank you so much for listening.
Thank you very much. We have time for some questions. Let me start with a question. These differences in FOT1 protein levels,
yes,
are they already visible in your triple mutant if you look at etiolated seedlings? Because I think all of these measurements you made is obviously done in light-grown plants,
yes.
right?
They're 14-day-old plants. Okay, so I think this can also be observed in etiolated seedlings. As a matter of fact, last Tuesday, Sakai-san was the one who presented data about PRR579 triple mutant, and I believe he was using etiolated seedlings during in his experiments, and they're showing that phototropin 1 is also overly expressed in the 579 triple mutant, and that it also affects phototropic curvature in plants.
Thank you very much.
Thanks for your talk.
Thank you.
Can you maybe say a little bit about what you think the physiological significance is of circadian gating of the chloroplast movements?
Okay. So I think the main biological significance would be is that, again, it optimizes the photosynthetic efficiency during morning conditions. As we can see from my results, chloroplast movement, specifically accumulation response, significantly enhanced in the 579 triple mutant, but that cannot happen if there's no, without the regulation of the PRRs, this would continue, and this could, of course, affect photosynthesis, specifically by damaging the photosynthetic machinery at high light conditions.
I guess I was wondering more, is there differences in the timing of the gating, say, for accumulation and avoidance? In other words, perhaps you might have peak sensitivity for accumulation at one time of day, but peak sensitivity for an avoidance response at a different time of day. Have you looked at that?
I've tried ZT0 and ZT4, and the difference that I'm getting, although I'm getting more accumulation response at ZT4, the difference in comparison to ZT0 is not that striking for me, at least.
I just had a question about FOT2. So have you looked at FOT2 levels?
Yes, I have data about phototropin 2 concentration. Clearly, phototropin 2 is somehow being also being influenced by the PRRs. But I also would like to emphasize in this figure that compared to phototropin 1, the rhythmic expression patterns of phototropin 1 seems to be more severely altered. in the PRR misexpressing lines when compared to the rhythmic expression patterns of the phototropin 2 receptor. So with that observation, I think that PRRs have more significant impact towards phototropin 1 compared to phototropin 2.
Okay, so I think that answers my second question. So the impacts on chloroplast avoidance movement
Yes.
were less than those in terms of accumulation.
Accumulation
So,
response.
So if you've got a more higher level of FOAT1 compared to FOAT2, you're sustaining the accumulation.
Yes, that is our, considering that phototropin 1 and phototropin 2 have this antagonistic nature towards each other.
Okay, thank you.
Thank you.
Thank you very much for this fascinating talk.